Welcome to Biochemistry Decoded, your ultimate guide to all your biochemistry doubts. Simplified and decoded. Today's topic is on acid-base balance, a must-know topic for every healthcare professional. Whether you're a med student, nursing student, or preparing for exams, understanding this concept is critical for interpreting blood gases and managing patients. Imagine your body as a tightrope walker, balancing pH between 7.35 and 7.45 to keep you alive. Too acidic. Too basic. Either way, it's trouble. In this masterclass, we'll break down how your body uses buffers, kidneys, and lungs to stay in that sweet spot. By the end, you'll master blood buffers, renal regulation, and respiratory control, high yield for exams and clinical practice. Let's start with the basics. pH is a measure of hydrogen ion concentration. Acids donate hydrogen ion, bases accept. So in an acidic solution, hydrogen ion concentration is more than hydroxide ion, and in a basic solution it's vice versa. Now, the normal blood pH is between 7.35 to 7.45 and the pH sustainable to life is between 6 to 8. In order to understand how acids and bases behave in solution or in our body, it is important to explore the Henderson-Hasselbalch equation. So, what exactly is the Henderson-Hasselbalch equation? At its core, it's a mathematical relationship that connects the pH of a solution to the properties of a weak acid and its conjugate base. Here's how it looks: pH equals pKa plus log A / HA. In this equation, pH is the measure of acidity or basicity of the solution. pKa is the acid dissociation constant, a unique value for each weak acid that tells us how easily it donates a proton. A is the concentration of the conjugate base, and HA is the concentration of the weak acid. The log term compares these concentrations as a ratio. This equation is especially useful for buffer systems, solutions that resist changes in pH. To understand where this equation comes from, we start with the dissociation of a weak acid, HA, in water. It partially breaks apart into H and A, its conjugate base. This process is described by the equilibrium expression, Ka equals HA / HA. Here, Ka is the acid dissociation constant. Now, if we take the negative logarithm of both sides, because chemists love working with pH, we get log H equals log Ka plus log A / HA. Since log H is pH and log Ka is pKa, this simplifies to the Henderson-Hasselbalch equation, pH equals pKa plus log A / HA. It's a beautiful shortcut that skips complex calculations and gets us straight to the pH. Why does this matter? The Henderson-Hasselbalch equation shines when we're dealing with buffers. It helps in 1. Calculating the pH of a buffer solution. By knowing the pKa of the weak acid and the concentrations of the acid and its conjugate base, you can calculate the pH of the buffer. 2. Understanding buffer capacity. The equation helps understand how a buffer solution resists changes in pH when small amounts of acid or base are added. 3. Preparing buffer solutions with a specific pH. The equation can be used to calculate the required ratio of acid to conjugate base needed to create a buffer solution with a desired pH. The Henderson-Hasselbalch equation isn't just theoretical, it's everywhere. In medicine, it helps explain how our bodies regulate blood pH. In pharmaceuticals, it's used to design drugs that work best at specific pH levels. Even in environmental science, it helps us understand how soil or water resists acidification. This equation bridges chemistry and the real world. Let's talk about pH again. The pH scale measures how acidic or basic a solution is, ranging from 0 to 14. A pH of 7 is neutral, below 7 is acidic, and above 7 is basic. Our blood, however, must stay slightly basic, within a narrow range of 7.35 to 7.45. Even a slight deviation can disrupt vital processes, so the body has evolved remarkable systems to maintain this balance. And a blood pH of 6.8, sustainable to life. But where do acids and bases in our body come from, and how do we keep them in check? Let's start with the sources of acids. Acids are substances that release hydrogen ions in solution, lowering pH. The body produces acids naturally as a byproduct of metabolism. One major source is carbonic acid, formed when carbon dioxide, a waste product of cellular respiration, combines with water in the blood. This reaction, catalyzed by an enzyme called carbonic anhydrase, happens constantly as our cells burn glucose for energy. Another significant source is lactic acid, produced during intense exercise when muscles work anaerobically, meaning without enough oxygen. Lactic acid builds up in muscles and spills into the blood, temporarily lowering pH. Other acids, like keto acids, can form during fat breakdown, especially in conditions like starvation or uncontrolled diabetes. Even digesting protein-rich foods generates sulfuric acid and phosphoric acid as amino acids are metabolized. Now, what about bases? Bases, or alkalis, accept hydrogen ions, raising pH. The primary base in the body is bicarbonate, a key player in our blood's buffering system. Bicarbonate is produced by the kidneys and, to a lesser extent, by the pancreas, which releases it into the digestive tract. Bases are less abundant than acids in the body because our metabolism naturally leans toward acid production. But don't worry, our body has clever ways to balance this. Buffers as your body's shock absorber. When acid or base levels spike, buffers jumps to stabilize pH before things go haywire. Body uses blood buffers, kidneys, and lungs to stay in that sweet spot, maintaining the pH between 7.35 to 7.45. Chemically, buffer is a mixture of weak acid and a salt of its conjugate base or vice versa. Its main function is to resist changes in pH when a strong acid or base is added to the solution. Our body has four major blood buffers: bicarbonate, phosphate, protein, and hemoglobin. These work in plasma, cells, and even our bones. Primarily located in the extracellular fluid, particularly the blood plasma, the bicarbonate buffer system is the most significant buffer in the human body. The system effectively neutralizes excess acids or bases, maintaining the blood pH around 7.4. Is our body's first line of defense against pH changes, the most common extracellular fluid buffer. It maintains a delicate balance between carbonic acid and bicarbonate, backed by the lungs and kidneys. So, how does it work? At its heart is this chemical reaction, carbon dioxide plus water forming carbonic acid, which splits into hydrogen ions and bicarbonate ions. In healthy blood, bicarbonate levels are about 24 milliequivalents per liter, and CO2's partial pressure is around 40 millimeters of mercury. This creates a 20-to-1 ratio of bicarbonate to carbonic acid, keeping pH at 7.40. This reaction isn't slow, it's supercharged by an enzyme called carbonic anhydrase, found in red blood cells. It speeds up the process thousands of times, making the buffer system incredibly responsive. The lungs are key partners here, controlling CO2 levels, and kidneys handle bicarbonate. The detailed mechanism of which will be discussed in the subsequent slides. So let's see how does bicarbonate carbonic acid buffer system works. First, handling acidosis. Let's see it in action. Imagine you're sprinting, producing lactic acid, or you're in diabetic ketoacidosis with ketone bodies. Your blood pH drops below 7.35. Bicarbonate ions act like a sponge, soaking up excess H+ to form carbonic acid. This carbonic acid breaks down into CO2 and water, and you exhale the CO2 through your lungs, raising pH back toward normal. Next, handling alkalosis. What about when blood gets too alkaline, say above 7.45, maybe from vomiting, which loses stomach acid? Carbonic acid steps up, releasing H+ ions to neutralize the excess base. Extra bicarbonate ions are then excreted by your kidneys to restore balance. The phosphate buffer system, while a minor contributor in blood plasma, is a vital component of the body's acid-base regulation, particularly in intracellular fluid and renal tubules. Its ability to buffer hydrogen ions and support acid excretion in the kidneys makes it indispensable for maintaining pH homeostasis. The phosphate buffer system consists of two key components: sodium dihydrogen phosphate, which acts as a weak acid, and sodium monohydrogen phosphate, which acts as a weak base. The pKa of this system is approximately 6.8, which is close to the physiological pH of 7.4, making it effective for buffering in environments where pH fluctuates around this range. Let's see how it works in action. When blood becomes too acidic, say, from lactic acid during intense exercise or ketones in diabetic ketoacidosis, monohydrogen phosphate steps in to bind excess hydrogen ions, forming dihydrogen phosphate. This reaction can be written as, hydrochloric acid reacts with disodium phosphate gives monosodium phosphate and sodium chloride. Here, a strong acid like hydrochloric acid is converted into a weaker acid, minimizing the drop in pH. Conversely, if blood becomes too alkaline, perhaps due to excessive vomiting that loses stomach acid, dihydrogen phosphate donates a hydrogen ion to form monohydrogen phosphate, producing water and stabilizing pH. That reaction looks like, sodium hydroxide reacts with monosodium phosphate to give disodium phosphate and water. In this case, a strong base like sodium hydroxide is converted into a weaker base, preventing a significant rise in pH. The protein buffer system relies on the unique properties of proteins, which are made up of amino acids. Amino acids have amino groups that can act as weak bases and carboxyl groups that can act as weak acids. Certain amino acids, like histidine, have side chains that are particularly effective at accepting or donating hydrogen ions, H, making proteins versatile buffers. When blood becomes too acidic, say, during intense exercise producing lactic acid or in diabetic ketoacidosis with ketone buildup, there's an excess of H ions. Proteins, especially those with histidine residues, bind these H ions, reducing acidity and stabilizing pH. For example, the imidazole group in histidine has a pKa around 6.0, which is close enough to physiological pH to be effective. Conversely, when blood becomes too alkaline, perhaps due to vomiting, which loses stomach acid, there's a shortage of H ions. Proteins can release H ions from their carboxyl or side chain groups, lowering pH back toward normal. The protein buffer system is a powerhouse, contributing about 2/3 of the buffering capacity in blood and most of the buffering within cells. Blood plasma, albumin and globulins are key players. Albumin, a major plasma protein, has multiple binding sites for H ions, making it a significant buffer. One albumin has 16 histidine residues. However, the star of the protein buffer system in blood is hemoglobin, found inside red blood cells. Hemoglobin is a multitasking marvel. Beyond transporting oxygen, it's a critical buffer during carbon dioxide, CO2, transport. When CO2 from tissues enters red blood cells, it combines with water to form carbonic acid, H2CO3, which dissociates into H and bicarbonate, HCO3. Hemoglobin binds these H ions, preventing a drop in blood pH. This process is enhanced by the Bohr effect, where H binding to hemoglobin promotes oxygen release to tissues. Hemoglobin also forms carbamino compounds with CO2, further aiding acid-base balance. The lungs' role in acid-base balance centers on their ability to control the partial pressure of carbon dioxide, or PCO2, in the blood. CO2 is produced by cellular metabolism and combines with water to form carbonic acid, H2CO3, which dissociates into bicarbonate, HCO3, and hydrogen ions, H. The amount of CO2 in the blood directly affects carbonic acid levels, which influences pH. The lungs regulate PCO2 by adjusting the rate and depth of ventilation, how fast and deeply you breathe. This process is governed by central chemoreceptors in the medulla oblongata and peripheral receptors in arteries, like the carotid bodies and aorta, which sense changes in pH, partial pressure of carbon dioxide, and oxygen levels. The lungs' buffering action is rapid but has limitations. They can only adjust volatile acids like carbonic acid, which gets converted to carbon dioxide, not bicarbonate or non-volatile acids like ketones. How the lungs respond to pH changes. When blood becomes too acidic, example from lactic acid buildup during intense exercise or ketones in diabetic ketoacidosis, the pH drops below 7.35. Chemoreceptors detect the increase in hydrogen ion or carbon dioxide and signal the respiratory centers to increase ventilation, hyperventilation. This causes more carbon dioxide to be exhaled, reducing partial pressure of carbon dioxide. Lower carbon dioxide shifts the equilibrium to reduce carbonic acid, decreasing hydrogen ion concentration and raising pH back toward normal. Conversely, when blood becomes too alkaline, perhaps from hyperventilation due to anxiety or high altitude, the pH rises above 7.45. Excessive carbon dioxide loss reduces carbonic acid, lowering hydrogen ion levels. Chemoreceptors may slow ventilation or hypoventilation that retains carbon dioxide, increasing partial pressure of carbon dioxide and carbonic acid, which releases more hydrogen ion to lower pH. Ever wondered why urine pH is typically lower than plasma pH? That's because kidney actively excrete hydrogen ion into the urine, making it more acidic. This process helps regulate the body's acid-base balance. Let's dive more into it. The kidneys are the body's long-term pH regulators. They do this through 4 mechanisms, all happening in the nephron, the kidney's functional unit. These are bicarbonate reabsorption, bicarbonate generation, ammonium excretion, and titratable acid and other excretion. These processes, driven by the nephron's proximal tubules, distal tubules, and collecting ducts, ensure a robust acid-base balance. Mechanism 1, bicarbonate reabsorption. The kidneys filter about 4,000 milliequivalents of bicarbonate daily, and they reabsorb nearly all of it, about 80 to 90%, in the proximal tubule. Here's how it works. Filtered bicarbonate in the tubular lumen combines with hydrogen ions secreted by tubular cells to form carbonic acid. The enzyme carbonic anhydrase, present in the tubular cells and brush border, converts carbonic acid into carbon dioxide and water. Carbon dioxide diffuses into the tubular cell, where carbonic anhydrase reverses the reaction, forming carbonic acid, which dissociates into hydrogen ion and bicarbonate. The bicarbonate is reabsorbed into the blood, while the hydrogen ion is secreted back into the tubule to repeat the cycle. This process preserves bicarbonate, maintaining blood's buffering capacity. In metabolic acidosis, when pH drops below 7.35, reabsorption ramps up to restore bicarbonate levels, typically around 24 milliequivalents per liter. Mechanism 2, hydrogen ion excretion and bicarbonate generation. Sometimes, bicarbonate is depleted, like in diabetic ketoacidosis, where it is consumed by buffering excess acids. The kidneys generate new bicarbonate to replenish it, primarily in the proximal tubule and collecting ducts. This process is tied to hydrogen ion excretion. When tubular cells secrete hydrogen ion into the lumen, they produce bicarbonate inside the cell as a byproduct, which is then transported into the blood. This new bicarbonate helps restore the blood's 20:1 bicarbonate to carbonic acid ratio, stabilizing pH. Generation is crucial in chronic acidosis, where the kidneys work overtime to compensate for ongoing acid loads. Mechanism 3. Ammonium excretion. The kidneys also excrete hydrogen ion in the form of ammonium ion, NH4, a process called ammoniogenesis, mainly in the proximal tubule. Here's the breakdown. Tubular cells metabolize glutamine, an amino acid, into ammonia, NH3, and bicarbonate. The ammonia diffuses into the tubular lumen, where it binds with secreted hydrogen ion to form ammonium ion, which is excreted in urine. This removes hydrogen ion from the body and adds new bicarbonate to the blood, boosting pH. In acidosis, glutamine metabolism increases, producing more ammonium ion, up to 200 millimoles per day compared to a normal 30 to 40. This mechanism is vital in conditions like renal tubular acidosis, where acid excretion is impaired. Mechanism 4. Titratable acid and other acid excretion. The kidneys also excrete hydrogen ion as titratable acids, primarily dihydrogen phosphate, in the distal tubule and collecting ducts. Filtered monohydrogen phosphate in the tubular lumen binds secreted hydrogen ion to form dihydrogen phosphate, which is excreted in urine. This process removes hydrogen ion without depleting bicarbonate, as each hydrogen ion excreted is paired with a new bicarbonate generated in the tubular cell and sent to the blood. The phosphate buffer's pKa of 6.8 makes it highly effective in the acidic urine environment, where pH can drop to 4.5. In addition to phosphates, other weak acids like creatinine and urate can contribute to titratable acidity, especially when the urine pH is low. Keto acids and lactic acid can also be excreted by the kidney, particularly during pathological conditions. That's acid-base balance in a nutshell. We covered the bicarbonate, phosphate, protein, and hemoglobin buffers soaking up hydrogen ion. We saw lungs adjust carbon dioxide in minutes and kidneys fine-tune hydrogen ion and bicarbonate over days. These systems keep your pH rock steady. Hope you enjoyed learning. I have also provided you all with a set of problems and solutions to assess your understanding on the topic. Kindly go through it. Want more high-yield content? Subscribe to Biochemistry Decoded for more medical biochemistry breakdowns, hit the bell icon, and do drop a comment. What topic should we tackle next? Let's keep learning.